Simone Pengue is a biophysics PhD student at Biozentrum of the University of Basel. He is also a freelance science journalist and has recently released his first documentary, Forgotten Data, The Leftovers of Science, which granted him the Prix Media Newcomer 2021 prize. During his talk, we will learn more about his documentary and how to uncover the invisible to this topic. Thank you, Simone, for being here.
Thank you so much for the nice introduction and for inviting me, of course. Do you see my screen? Do you see my slides, right?
Yes.
Okay. So let's just put this a bit in a context to begin. First thing, as you said, I'm a biophysicist and also like in my spare time, let's say I'm also a science journalist. And here I'm trying to combine the two things, bringing to the general public the needs of a scientist in the field of molecular biology. When we talk about data, open data, it's very important to put it in a context, to say in which field we're talking about. And we'll also see this in more detail later. And talking about data in molecular biology means that, first of all, you have a lot of diversity in the data you're talking about. And second of all, there is no privacy concerns. There is nothing linked to people. And this is, I think, very important to distinguish the data that we'll talk about today from many things that we've heard today in the previous talks. Now, I also know that this is the last talk of today, and I also know that everyone is tired, of course. So let me just keep it simple. And we're going to talk about protein dynamics in nuclear cytoplasmic transport. And this is my project. Well, actually, we're going to simplify it a bit. And instead of talking about proteins and lipids and lasers, we actually just going to talk about monkeys going left and right in a forest. Just want to introduce my project because you will understand why. So the goal of my research is to study the motion of these monkeys or these proteins. And these monkeys are jumping from one tree to the next tree. How do we study them? We just hear their voice because these are screaming monkeys. So we just hear their voice, we follow their voice, and we try to understand how fast they're moving. So what we found out, and this is our result of our research, and that no one is interested in it right now, I know, is that the speed of the monkey increases with the number of monkeys. And that's beautiful. And this is our result. This was the goal of our research. It was the reason why we got public funding to do this research. But while I was doing this research, while I was in the lab acquiring the data, I actually noticed that in the meanwhile, it was not just the monkey speed was increasing, but the pitch of their voice was changing, was actually decreasing. Well, in the real science, this was the lifetime of the fluorophores, but yeah, this is not important. So what's the key message that I want to bring here is that while I was looking for something, I also noticed there was something else in my data that I was acquiring anyway. So I was super excited and I showed this to my professor and he told me, yeah, beautiful. And so what? So the idea is that we had an answer, but we didn't really have a question. And at this point, I started thinking, okay, but I anyway have this data and we spend a lot of money and a lot of time and a lot of energy to generate this data. But what is going to happen to this? As a scientist, I'm just told, put this data in a server, but then is anyone actually going to access it sooner or later? Is anyone going to do new science with the data that I acquired anyway? And these were the questions that came to my mind when I really faced this problem for real. So where does all this scientific data actually ends up? We put this on the server, but we don't know what's happened to them in 5, 10, 20 years from now. Can we recycle them? So can we make new science out of this data? And if we can recycle them, how and why? This is absolutely not clear to, I think, the most of my colleagues here in my institute, the Biozentrum. So the occasion was this Prix Media Newcomer, which is a prize from, it's a contest from the Science Academies, no, the Swiss Academies of Science, so which is an institution, a confederal institution that promotes let's say the science communication in Switzerland. and they're just founding the project. So I thought it was the perfect occasion, because if you want to do science communication, you need first thing money, second thing some distribution channel, a third actually in the context. If you want to interview someone, you just write an email, hey, can I interview you? These people are more willing to say yes if they know what you're interviewing them for. So this was the perfect occasion. And this guy here on the right, this is my brother. And he actually is the person who helped me doing the movie. He's the person who's able to handle the camera, to do the editing. And without him, all this would have not been possible. And I just reached out to him and said, do you want to do a movie? Do we have time this summer? He said, I don't have time this summer. I said, yeah, just find the time. We have to do this movie. That's it. And we just did it. And we will now see the trailer. So let me just try to do the magic trick once again. It worked when I tried it, and it actually worked. So let's see if I'm lucky enough. I should be sharing. Are you now seeing like a video? No, it's not started yet.
Yes, we see a black screen.
Perfect. So I'm gonna start it. This is the trailer, 1 minute trailer. Okay, let's go.
Ogni giorno nei laboratori di biologia svizzeri vengono prodotti enormi quantità di dati, come immagini al microscopio, sequenze genetiche o test biomedici. Una piccola parte di questi dati è effettivamente utilizzata, mentre la grande maggioranza sono invece messi da parte. Che cosa succede a tutti questi dati scartati? E soprattutto, è possibile riciclarli?
Yeah. Yeah.
Where we need to get to is where data management becomes sexy, you know?
Yeah.
Che un ricercatore pubblica i suoi dati, proprio i dati.
Yeah.
And so why not reuse what is there instead of spending more money to generate an initial data set?
Yeah.
Servers use electricity and sea levels are rising.
Yeah. Yeah.
Okay, so now stop sharing here and resume the sharing of my PowerPoint. Here we go. And so this was the trailer. I think it says a bit of what we're going to talk about and in which style, let's say. And so let's just go through the main points. What we'd like to bring here are some, let's say, in a sort of a summary of what I found out during the movie, and to include maybe a few things that I did not have the time, or in a sense, I did not want to put in for some reasons, and I'm just going to take the occasion to actually talk about this here. So why not reuse what is there? The person who said this sentence in the trailer was Professor Michele Zavolan of the Institute of Bioinformatics, and also here at Biocentrum from University of Basel. She's a professor in bioinformatics, and she's an expert in artificial intelligence and machine learning, and in general, we can say in data mining. So this is the reason why she took part in the movie. She explained us how to extract information out of large amount of data, which also we saw very much today in several talks just before mine. So why not reuse what is that? Or actually, why should we do it? First thing is a matter of efficiency. Doing lab research costs a lot of public money, but really a lot. I've heard that every scientist in Switzerland in the lab, like a biology lab, spends like 600 francs per day. And just in my institute, we are 500 people. And not to talk about many other money that can spend here and there. So getting the most out of this data actually would also make like sense economically. It's a matter of transparency. Science should be transparent. I should be able to see if someone is committing some fraud or if what they're claiming is actually true. I should be able to reanalyze it. And then if you actually are able to combine data set acquired from multiple people, the potential in terms of quantity of data is completely incomparable to what you can acquire by yourself. And this was pretty clearly stated by Roche. So here in Basel, there is, well, of course, you know Roche, the pharmaceutical company. And we interviewed them actually just in written form. They replied to some written questions. And we asked them, do you recycle your data? They said, yes, if we don't do this, it's a huge waste of time and money. We have to do this. And when we do this, we are able to actually have a much larger cases for our studies of our drugs. They said that only they test on people, for example, 10 different molecules to produce one drug. Out of these 10 molecules, 9 are discarded, only 1 is actually commercialized. And the data from the 9 other potential molecules, they are still used to better understand the disease and eventually produce different drugs for different, for example, diseases. And the data, the actual data. The person who said this is Maria Grazia Giuffreda. She's the Associate Director of the Swiss National Supercomputer Center, which is in Lugano, but is part of the ETH. She is a very, very much an open data and open source enthusiast. I actually suggest Daria to maybe consider inviting her for next year. She's also a very good speaker. And the first thing that you have to think about is that in science, especially in biology, paper figures, so what is published is not the actual data. That's a selection of very well polished data, but there are many, many more information hidden somewhere. Now, we're talking about recycling this data, but no one is ever going through my hard disk or the server of my research group, or the research group I belong to actually dig through my data, like randomly organized with random names and put all together just like a huge pile of garbage. Where people actually look for data are domain-specific repositories. So websites, basically, servers that contain data only relevant to one specific aspect of research. For example, protein structure, we'll talk about this. Or for example, I don't know, genomes, human genomes, data about cancer, cell data about cancer, these kind of things. It must be domain specific because sometimes, I mean, people don't really know where to look for things. The idea is very much like a library, like an ordinary library. If the books are organized randomly, you will never find the best book in the world, even if it's in there. And along with this idea of domain-specific repositories, there are metadata, data about data. These are really the key to be able to reuse and recycle the data. It's just like when we do recycling of garbage at home. We have to know what we have in our hands to put it in the right bin. This is glass, goes with glass, and the person who's gonna pick up my trash knows that there's only glass in there, and so they can actually use it. They can rely on this information and actually do something useful out of it, because there are information about the things that are in there. And it's the same thing with data. If you want to recycle data, if you want to reuse data, scientific data, we need very good annotation. And actually, of course, I'm not the first one who thought about this. And it's a big thing in science, especially in biology and molecular science. And in 2016, the scientific community, so it was a cooperation of institutions and scientists, published these FAIR principles. I don't know how many people are actually familiar with these FAIR principles. It are some guidelines for scientific data to actually promote the reuse and recycle of this data. A simple guidelines that don't provide any service. It's really just how you should put your data out there. So first thing, your data should be findable. So put there in a position where people can actually find them, and most importantly, put annotation, put metadata, so that people know what this data have inside. Make them accessible. This, I think, is self-explanatory. Make them interoperable. Okay, I think that they couldn't find a better word with the letter I. The idea is that this data should be readable by humans and by machines. meaning that they should be in a standard format. They should be organized properly in such a way that you can just feed a machine with this data and do eventually machine learning. So they should respect some sort of standard, depending on the domain, of course, because you said they should go in domain-specific repositories. And they should be reusable. If you respect all these things and your data are good and they are reliable, someone else can actually do good science out of it. Sexy data management. We have to get to the point where data management becomes sexy. The person who said this in the trailer is Michael Podvinets. He's the head of technology platform. He's by training is a microbiologist and very much into IT. And he's actually the person who originally inspired my idea of talking about these things. And he's also very knowledgeable in terms of data management. One of the key messages that he's trying to deliver is that we have to bring out there many successful stories of data recycling. We have to tell people, scientists, for example, that other people did great science with data produced by other labs, with just recycling of data. So here I thought about bringing this example of AlphaFold. AlphaFold is a program, let's say, from DeepMind, which is a company owned by Google. So this is really an exchange between the public and private research. And this AlphaFold is the largest revolution in molecular biology of the last maybe decade. I don't know. It's a huge revolution that just came out a few months ago. Everyone is talking about this. So what does AlphaFold do? AlphaFold predicts the protein structure. What is protein structure? Now, the protein, you know, they are the basics of life. All the cells are made of proteins. The proteins are made of, we can imagine some small Lego bricks, different colors, and they just do the sequence of the protein. So we know that there's the red brick followed by the yellow, followed by the green, followed by the blue, and they just form a line. This is the protein sequence. Now, to actually understand how the protein works, a key information to have is the protein structure. So how, like the three-dimensional structure, to understand how this object, this item works. If I know that it's made of glass, I don't have all the information. I need to know the shape to be able to understand that I can fill it with water and it's just a bottle. It's a bit the same thing. With proteins, we need to understand how they're made. Actually, going from the sequence, so just knowing that it's a red followed by yellow, followed by blue, green, red again, to the structure is a super complicated problem. The computationally speaking is something that people are trying to solve since decades. And now AlphaFold, using artificial intelligence, managed to predict successfully, like with a reliability of like more than 90%, the protein structure starting from the sequence. Now, I took this sentence, I think it was very strong, from their blog. We trained the system on publicly available data consisting of 170,000 protein structures from the Protein Data Bank, which is a public repository, domain-specific, together with large databases containing protein sequences of unknown structure. So what they did, they took a huge amount of data, publicly available, and this data is maybe not published in paper. So maybe this data is something that someone generated like on the side to do some other research. And they took all this data and they put it together with some other data, which by itself had little value. And they managed to do the largest revolution in structural biology. Just feeding a machine, basically, because to feed a machine, you need a lot of data. We also heard this earlier in the talk by Professor Salatin. And then sea levels are rising. This is David Shaw, a researcher. He's an expert in biomedical ethics, and he's at the University of Basel. And the idea is that, yes, there are many reasons why we would need to share data, but there are also many reasons why maybe we should slow down our enthusiasm in a sense. Actually, it's not that everything can be shared. It's not that everything can go out there. Yesterday, I took some microscope pictures. They were disgusting. They were completely out of focus, and the conditions that acquired them was super dirty. That data maybe should be forgotten. because keeping it in some server would be wasteful. It consumes electricity, it consumes space, it's public money, and it's, I mean, it's also, it's really a challenge. So the idea is some data must be forgotten. I was discussing with a professor here at the Biocentrum, and he told me, after he watched the documentary, and then he reached out to me and he said, well, actually, I think that some data must be forgotten because it's exactly what happens every day in life. When we just live, like walk around, we are surrounded by data that come to us in every single moment. What we see with our eyes, what we hear with our ears. These are all data, and our brain is actually so good that can discard the vast majority of it and only keep the crucial information. And a scientist, in a sense, has to do a bit the same thing. Just know where the crucial informations are and discard everything else. And this is the first concern, in a sense. Then the big, one of the main things that's creating viscosity in the field when we talk about open data and shared data is that there is no real common agreement on intellectual property. A few days ago, I was talking with my colleague here, and she showed me some data. So I was getting this talk ready, so I had these things in the back of my mind. And she was showing me this data. She said, well, this data is interesting, but it's not enough to do a publication. And then I said, yeah, why don't you share it with someone else? Why you put it on some public repository or something like this? And she said, yeah, but then would I be an author or would just be acknowledged? I spent time on this. And if I'm an author, then I want to be able to edit and edit the paper, edit publication, edit the sentence written there, because that's my data, that's my signature on it. If I'm not an author, then I don't see why giving my effort to these other people who's going to take all the credits. So there is no common agreement. People don't share the same opinion on like how we give properly credits for data. There might be privacy concerns, yes. I said this is molecular biology and cellular biology, so there are nothing that directly links to people, but there might be. So this is always something that we have to keep in mind in general. And then very importantly, one size does not fit all. Open data, data sharing is something that's extremely domain specific. I'm not only talking about molecular cellular biology and so on. I'm really talking about the entire science. What I said today, it really only holds for cellular and molecular biology. If we talk about high energy physics, for example, no, that's miles away from what I said. They share everything by default. I have friends that base their entire PhD research on reanalysis of data acquired by something else. That's standard. Well, these people invented the internet, so what can we expect from them? Of course, they're sharing everything. And if we change, we go to medicine, where there are, or fields where there are people's data involved, the privacy concern becomes much stronger. Of course, we already discussed this today. And if we go to other fields, for example, literature, I do also some research in literature, and there you cannot even access your own paper. So the idea of sharing the data, it's a complete fantasy. And there are many, many shades of gray in between. It's really something that depends a lot on what you're doing. If you're doing studying with people, maybe these people do not want their data to be shared, but if you ask, can I share your data, then you're biasing your sample, for example. So it's really hard to find a solution, especially for policymakers. So perfectly in time, we are getting to the conclusion. For the institutions, we need coordination. Data recycling works very well at Roche, works very well inside individual research group because there is a coordination. That's what we need. The discussion must be kept alive. Yes, we have to discuss everyone. We just can't someone else will take care of it. But most importantly is that we need to inform, educate, and spread information or concerns about data sharing. Me as a scientist, I did a movie on this, okay, and I still don't know where to put my data. That's ridiculous. People are not informed. Some other people, some journal asked me to put some repositories. I don't know really where it went. Someone else took care of it. I mean, there's no education on data sharing among scientists. For the scientists, first thing, share your data. Don't be afraid. Don't be shy. but also use shared data. We need to produce successful stories, sexy data management. So use shared data and write on your paper that you use shared data. But most importantly, this is something that we have to do all together. It's a collective project. We need to come up with needs, troubles, concerns, whatever I'm saying that someone disagrees with, please just say it in this forum or via email to me or to your friends, to someone, but just say it loud, because the potential is so big that you really need to start a constructive discussion on this, and we need to know each other's needs. And this was a bit the conclusion. I just wanna spend the last 30 seconds. I have the timer here. On doing some acknowledgements. So first thing, my brother Lorenzo, of course, he did. He really did half of the job, and so the other 4 people here, they actively supported me. Laura Marino, Nicole Maniero, Florian, and Elsa. They really worked on my side all the time. All the institutions that actually supported me. And then I really want to thank Darien and Opendata.ch for inviting me. That was really a great opportunity. Thank you.
Thank you so much, Simone. And what an exciting trailer of your documentary. I'm sure many of the people here will watch it afterwards. Do we have any questions? The one that is very obvious, you've mentioned it yourself. So what are you doing with the data currently that you create during your research?
That's a good question, because it does not depend only on me. I'm a PhD student. I have to, if I want to share it, if I want to put it on some public repositories, first I have to ask the permission of my PI. And now, what's his opinion on open data? Well, I don't want to speak with him for him, and so what I would like to do is, for example, put some data on some domain-specific repositories, right? What could be this repository? Could be something that, for example, people share data about molecular diffusion. I had some needs. I wish I was able to access some type of data, and then know that there is no repositories in the domain that I work in. So I just hope that maybe discussing with someone today or in the next days, we can come up with a solution to share data about molecular diffusion, for example, or for behaviors in general. If someone knows where I can put this data, please just send me an email. So what I'm doing with my data? Nothing, because I don't know what to do. Literally.
So that's a call for help.
In a sense, yes.
To the audience. We have a question or comment here. One thing that surprised in AI is that algorithms can be pre-trained on very general data, and then it gets a lot cheaper to train them on the more special cases. That might also be an argument for data reuse.
Yes, but remember, well, I'm not an expert on artificial intelligence. I agree. I think that this is also what we saw earlier with the food, right? With the food recognition. Let's start with recognizing food that is easily recognizable. Pizza, for example, I think that's the easiest. And then we get to Indian food, just one step at a time. But the thing is that when you want to train an artificial intelligence, you need a lot of data, really a lot. You wanna train your algorithm to, for example, recognize distinguish some type of cell from some other type, or I don't know, cancer cell from non-cancer cell in some specific condition. Okay, you need hundreds of thousands of pictures of this. Where do you take them? You will never be able to produce them yourself. And if you produce them yourself, you're introducing a huge bias, because maybe you don't know, but there is some sort of experimental condition that you are going to put in the data that is really biasing a lot. So you need to do artificial intelligence, or to train your machine in biology, in science. You need people from other data. So I mean, thank you for the great talk.
So thank you. We have another question with a long comment supporting what you've just said, and she recommends to train more young researchers in the domain of bioinformatics to make them less dependent on groups like Michaela Zawoland's lab. Apart from this comment, she's wondering if you're also in favor of publishing negative data.
So I think that by negative data, you are referring to, for example, it's a bit of an interpretation. I guess negative data is when you try, for example, you want to understand the best strategy to achieve some result. So for example, the best strategy in my case, you have the protein, okay, you have another molecule, which is a fluorescent molecule, it's going to bring the light, but this is the voice of the monkey, and this is the monkey. You have to put the two together. Okay, how do you put the two together? There are many different strategies. So what you usually do is that you just take, I don't know, 100 or maybe 20 different little tubes, and you do a very little condition, you change the condition, chemically speaking, from one to the next, a little bit, a little bit, a little bit, and you see which one is working the best. This is screening. Everyone does it in science all the time. Now, I think that what here Muriel is saying as negative data is like, okay, out of this maybe 20 tubes, you select one. What about the other 19? The thing is that these other 19, you cannot publish them in papers because, yeah, who cares? But maybe they're going to save a lot of money, a lot of time, a lot of energy to some other people. So yes, I would be in favor of publishing them, for example, because it would save my time as a researcher. If I know how to, if I have to do some test on the best strategy to achieve something like biologically or chemically, I would be very happy to see some other people test. Maybe we tested the same thing and we both saw that it just doesn't work. So yes, I would be in favor. Publish your data on zenodo.org and it's not easily findable as topic-specific repository. Yeah, this is the problem. So I didn't know this zenodo.org. So thanks. I'm just gonna save the link. Just open. Okay, so now I have on Chrome. Thank you. And link to your scientific publications. But of course, it's not easily as fundable as a topic-specific repository. This is the problem. Also, like the Swiss scientific, the Center for the Supercalculus in Lugano, in Italian is called Centro Svizzero Calcolo Scientifico. They also offer this long-term data storage. They keep your data safe for more than 10 years, and they give you a PID, persistent identifier, so that you can just have this address for your data. You can share this and say, everyone can just go there and directly download your data. You just need this address. So they do this service. I didn't know. So do I have time for a story? No, I don't have to do time for a story.
Yes, we have.
I have time for a story. When we went to this CSCS.
We have one more question.
Okay, thanks. When we went to this CSCS, we really, we had no idea that we were also doing data storage. And we went there only to film nice computers. So I wrote them an email and said, I'm doing a movie about computers and data recycling. Can we come there and just film your damn big computer? And when we went there, and we just started talking with Maria Grazia Giuffreda, and she said, no, but we're actually very active on open data. We are huge supporters of open source, and we also offer data storage services. And we just give this PID, which is a very important source for data sharing. So she was exactly like the key that I was looking for. Where do this data actually end up? They end up in Lugano, just in front of the football stadium. They go there. Many Swiss scientific data, they just go there, and I had no idea. So this is exactly what I said in the conclusion. We need to inform, we need to educate, we need to spread the word about how to properly do data management.
One more question from Wood. We forgot the model of libraries was the system of knowledge management until now. What do you think? How can we as a society recreate the same structures for data like we have in the librarian model?
Yeah, exactly. First thing that I wanna, so there are many aspects from which I can take this good question. First thing that it's not completely true that we forgot. the model of libraries, in the sense that there are actually data librarians as a profession, also in Switzerland. So this thing that the name stayed the same, I find it very, very intriguing. It means that the tradition is always the same. So how can we recreate the same structure for data? Well, first thing, it's we need infrastructure to do so. As we needed large rooms to be libraries, now we need large servers to be data libraries, and we need to organize our data. We need metadata. We need agreement on which metadata is important. How do we organize it? And this is really complicated. It's really complicated because in science you do, by definition, something that no one has ever done before. So the information that I can think they are relevant for my research, maybe in 10 years, they're going to be completely useless. While when we talk about books, it's a bit more standard the way that you can track the book. For example, you just choose, okay, I'm just going to divide everything by author name in alphabetical order, and that's it. And that's going to be it also in 1,000 years. But if now I divide everything by, I don't know, microscopy technique, for example, that's completely useless in a few years because the microscopy techniques are going to be so different. And who's going to care if I use turf or FLIM or whatever else in 10 years? Maybe, I don't know. We just don't know by definition of science. So how can we as a society create the same structure for data? I think this is the big challenge now in data management. It is, and now it's the right time to solve it. Other people do it in other fields, for example, high energy physics, where everything is much more quantitative. And you can just put numbers. In biology, in nanoscience, you can't do this. But we need some agreement, otherwise it's just too late. It's just like if you just start putting random files on your desktop. After a while, you have so many things in there that you're never gonna reorder it. You need to do it as the earlier you start doing it, the better it is. The very complex index card is kind of have books made by Norwegian for the index card. Yeah, indeed, but indeed, this is the thing that very complex index cards, I think they're more relevant for large libraries. It's exactly the idea of metadata. It's the same thing, data about data. So write text about the text about the book. And this is still the key now. Maybe
last question for today, I would say. Maybe we need guidelines which data are good for reuse.
The thing is that you don't know. So the picture autofocus that I acquired yesterday, are we sure that they are not good for use? Very likely. But how do you know? And this, I think this really, like, we need properly trained scientists who know what they're deleting, who know what might be relevant in a few years from now. If you don't know about your data, who's going to know it? Absolutely no one.